And the final construct, we had a his tag with the yellow fluorescent protein on the inside and the LC3 and emerald green on the other side. And it was expressed in VC21 cells and purified in chromatography. And when we did that, we found, of course, we were very pleased to find it was quite pure. It had the right mass, which was pleasant. And most importantly, when it was mixed with the enzyme, it was cleaved into the two halves. You could see efficiently that both halves were corresponding exactly to what expected. So it was not only you could make it, but it was a substrate. When we did the kinetics for this cleavage, we could see it was around 4 micromolar and behaved quite well and allowed us to set up a fluorescence assay. We could then look at this assay and look for inhibition with our chloromethylketone. Indeed, it did inhibit the enzyme cleavage of the substrate with a Ki around 1 micromolar. And equally, the fluoromethylketone also inhibited with a similar Ki. But when we look at this inhibition here, we find that there's about 6 of these molecules, the chloromethylketones, are attached to the protein when it's inhibited. Actually, ATG4B has 8 free cysteines, so in principle, any of those can react, although some of them are hidden inside. So that was not too good, and I often wondered when people use like N-ethylamide or chloromethylketones, maybe they're reacting with all sorts of irrelevant thiols which should screw up the protein. But luckily for us, and maybe by design to some extent, the fluoromethylketone in fact only reacts once, and it still inhibits. And we could show by mass spec that the ATG4B mass was increased by exactly what we expect from the alkylation of this one thiol. So in summary now, we have a rate assay using the LC3 substrate. We can measure it using mass spec if you want to use the actual LC3, but we've also made a fluorescent peptide assay, which can actually be of some use, although it requires a lot of enzyme. The fluorescent LC3 FRET assay has been developed and is now being worked up for high throughput screening by the CDRD and the DBC. And it's now in a 3 full plate format, and we hope to start screening very soon. We've also made a stable irreversible inhibitor, the fluoromethylketone, and it shows binds 1-to-1. And we're now just starting a crystallization of ATG4B NA with this inhibitor using soaking experiments and also some co-crystallization experiments. And then finally, in silico screening has Steve Jones and at the BC Cancer Research have selected compounds for screening. And actually, from these compounds, we've actually found an inhibitor already last week. And the pilot, as I mentioned, is going to go ahead. Now, I don't think I'm going to go through this. I'm just going to go through my, I don't think I'm going to go through this. I'll just go to my acknowledgements. And thank the people involved in these projects that are shown here. Some of them, David Bucado's lab was very helpful in getting us into the protein. And there are people at Toronto who are doing some of our bone experiments I didn't mention. CDRD people, of course, and Genome Sciences Center, Alan Edwards, and the social genomics people. And of course, BC and various other people who have been very important to support all this work. So thanks.
Thank you, Bob. And Bob has skipped some slides so we can have a discussion. So,
Yes, well, I thought it was.
Are there any questions for Bob? Dan?
Yeah. Jeez. I'm interested in the sociology of this. So how these collaborations are put together. Do you go to people? Do they come to you? Clearly, you could do bone as well as you could do pancreas as well as you could do brain.
I mean, some of them have come from personal interest. I'm interested in bone. So I went to Jane O'Dan, and she's an expert in bone. I said, would you like to collaborate? She said, okay. The autophagy actually came from a friend of mine who was interested in neurodegenerative disease, and he said, here's a target for a chief out for neurodegenerative disease, and I got interested in it. And of course, when I came here, I started seeing around people and I found that Sharon Gorski was very interested in autophagy, so we just got together and we see how we can synergize. And so you sort of find your way from either common interest or knowing people who know people. I guess one thing I find as a chemist is a very valuable adjunct to most projects, and many biological projects would be very much better to have chemical input. So there's a lot of people who are very interested in me to help. So I have to pick and choose. It's not a matter of sort of out of country.
Maybe as a follow-up, if I may, then maybe we go to the psychology after the sociology. So, I mean, what kind of mindset do you find works here to make these collaborations? Because you could just be in your own lab doing, structure analysis.
Yeah, I mean, I guess that comes from my background because I've always been driven to try and get results, meaning like meaningful results that will impact a wider audience, and in my case, sort of medical advance or advantage. And so, I'm not going to do that on my own. So I'm quite happy to orient myself to things that I think would be useful in the long term as therapies. It doesn't necessarily mean making drugs, but obviously finding new avenues to drugs. So, that's what drives me, but obviously it comes down to the interface of biology and chemistry. It's for me, a fundamental motivation, which implies you must collaborate. This is sort of can't be helpful.
Other questions for Bob? Well, I might just ask you to say one sentence or two around CDRD, because you're a critical part of CDRD. I didn't mention that at the start.
Yeah, I mean, I'm also part of the CDRD. I'm the head of the Division of Medicinal Chemistry there. It's made up of 5 different entities which span the various elements of drug discovery. And our idea is to have these capabilities to augment the efforts of academics who find various interesting targets, structures, compounds, whatever, but they're lacking the other bits, and so we can actually help to make people's projects move forward, or at least validate them, find out if they're flawed or not. And if they are worth doing, then we can perhaps supply them with the needed adjunct components. And so it's a bit like my own concept of collaboration. CDRD will actually screen for you, or they will engineer assays, or they will do formulation or animal studies. And so it's a very useful entity that I think is having an impact. And we should all think about that and have projects which are, you'd say, that's great. Unfortunately, I don't have a compound to test it, or I haven't had somebody to screen. CDRD would be a logical person or entity to entertain that possibility.
Thank you very much, Bob. We appreciate it.